So welcome back. This is the second lecture of the 4 series here on virus ecology and evolution. And just to remind everyone where I am in the sequence, the first 2 lectures will be on principles of virus ecology and evolution. I started, obviously, earlier in the week, focusing on single host virus interactions, and now I'll begin to expand. And as I mentioned before, on Friday I'll talk about continuation from lysis to latency, and on Saturday from theory to therapy. And again, as I mentioned before, I'm going to try to mix theory and modeling throughout, and I'll give probably more emphasis on empirical data and challenges today. Of course, there'll be theory mixed in and modeling, but I definitely want to give you a broader sense of the kind of empirical framework in which we're operating. And so to reiterate the 3 questions that I want to get through between the first lecture and today, first of all, how does viral infection change microbial population dynamics? And in doing so, that's really asking an ecological question. And then I'm going to try to make the link between ecology and evolution by asking to what extent, first of all, how does the ecology set up the potential invasion of a particular type in a new environment, which becomes an evolutionary question, but also then how evolution modifies population dynamics, and how does coevolution modify population dynamics. In doing so, I'll try to unite both ecology and evolution in the framework of eco-evolutionary dynamics. And then the last set of questions I'll get to today is to begin to probe this question of the relationship between virus-host interactions in a context of a complex community where there are many players, many viruses, and many hosts. And to do that, I have to first tell you a little bit more about what we know as a community about the interaction networks and what that might tell us about dynamics as well. Okay, so with my last lecture, I got to this point, so I'll just tell you the summary. The take-home message were there these models of virus-host interactions which led to the canonical predator-prey-like or lockable-terror-like population dynamics, where you have an offset between prey abundances and predator abundances, right? First the prey peak, and then the predator peak, and then we see the subsequent troughs. And so this gives us some information about which viruses are likely to invade. Just because a virus can infect a host at the cell level, it doesn't necessarily mean it's going to invade at the population level. But at the very end, I mentioned this problem, and I showed a little bit of data that evolution can rapidly change outcomes. So that's what I'm going to pick up today. Okay, so part 2 of this sequence, I'll try to answer this question: how does evolutionary change, and more broadly speaking, coevolutionary change, alter virus-host population dynamics? And I should point out that a critical part of this work was done with the former postdoc in my group, Michael Cortez, who's now an assistant professor of mathematics at Utah State, but he also bridges the gap between mathematics and ecology. Okay, so what was the data piece that I ended up with in the first lecture? It was, as you can see above, interactions between phage, which is a T4, which is an obligately lytic phage. It only is going to kill cells, and the host E. coli B. And the data is taken about 200 hours, and you can see these oscillations where you have blue for the bacteria, red for the virus, and you see the oscillations, and there's a time shift, so that slightly after the peak of the bacteria, you see the peak of the virus. Okay, so that was 200 hours. So I want to show you the continuation of this experiment out now to 600 hours. Right, so now we're going for almost a month in terms of time, and you can begin to see that something quite dramatic changes. And I'll just flip back and forth to show you and emphasize the fact that the former time series is just compressed in this part. But this is what happens thereafter. Right? And so there's a dramatic change here from what looks like predator-prey-like oscillations to something in which the prey dynamic seems relatively constant, and now the virus is oscillating. So first of all, this is, I hope, a bit unusual in the sense that we have a marked shift in dynamics. The other thing that I think should be unusual is the fact that if the hosts are constant and the viruses are oscillating, what's going on here? Right? Why isn't there also oscillations on the host side? And the third part that makes this particular plot a little bit mysterious is there wasn't any change in the experiment. So sometimes when I show this kind of slide, I like to ask the question, well, what has changed? We've given away. There's some evolutionary event that's taken place, right? But I want to emphasize the fact that there was no change in the external conditions, in the, let's say, the chemostat conditions in terms of a different concentration of resources, et cetera. So something intrinsic to the system has taken place. And there's been an evolutionary event. So now I'll at least ask that question. What kind of evolutionary event or what kind of event has taken place in this system? Any thoughts from students? So what do you think, what do you think has taken place here? Yes?
I have one opinion which is very wrong.
Okay, but it's an opinion, you can share it. Yeah?
They have stopped being predator-prey.
Stop being predator-prey. Okay, so we'll explore that point. Keep in mind that the viruses are obligate intracellular parasites. So if they stop being predator-prey, and I appreciate you making that suggestion, there's some element to which you will see that is borne out. This decline would make sense. They're no longer predator-prey. We have a prey, but it's no longer the predator. Except for the fact that the virus goes up. So it is infecting and replicating something. There's got to be some prey in that environment or it wouldn't go up. If we see the decline, that would explain it alone. Okay, so let me have some other thoughts. Yes?
I don't know what's going on, but phenomenologically, the host becomes constant only after, for the first time, the virus population falls below 7.
Right, so that's an interesting observation. I'm not sure, and I don't know if that's necessarily causal or if it's coincidence, but it is true that there was a drop there. I'm not sure that, I have to think more about it. I haven't thought to see if that's a hallmark. Maybe that's a hallmark of something. Yeah, there is an unusual feature there. But yes, let's see in the middle. Go ahead. So I'll show you another version of this experiment where they got very similar behavior. Okay, and also keep in mind there's a 200 hours later, the memory of the system in many ways has been forgotten, right, because of the turnover time in the chemostat. Okay, so we have some evolutionary event, one last try. I'm sticking with the students, just for, if I see your hand, but I'm intentionally ignoring it. Yes?
Maybe now we have like two populations of cells. One population is immune to the viruses, and the other one is highly susceptible.
Okay, so now we're poking away and getting much closer to where the action is happening. So there was some evolutionary event. There's been a suggestion that there's no longer a predator-prey dynamic, but you can see that we can reject that hypothesis because of this increase. But that doesn't mean necessarily that there isn't some truth to that comment, and maybe your comment should be explored. So in fact, that's what they thought. They thought that perhaps there was an evolutionary event, a mutation of the bacteria from sensitive to resistant. Let's use the word resistant rather than immune. I know we just had a lecture on immunity, but we're going to use the word resistance in this case. If I want to talk about immunity in bacterial cells, I'll talk about CRISPR. This is surface resistance. So now we have potentially a resistant population. Okay? So if there's a resistant population and a sensitive population and viruses, well then maybe there is an absence of a predator-prey relationship for a component, but not for the other. So the problem with this experiment is that they were not able to distinguish resistant and susceptible populations because they're just measuring colonies. So after this experiment was over, they did identify a resistant type, but instead of just tracking total population as we're doing here, they were able to introduce a marked resistant clone and track its dynamics separately from the susceptible clone's dynamics. And I'll show you what happened. So this is follow-up work in which...
Are there different strains of virus?
So there may be different strains of viruses, but they're not necessarily, let's say, we don't need to invoke them. I'm sure there are there, and having done these experiments, there are lots of viruses in the background, but we don't need to invoke them to understand what's going on, right? But we've done some work on coevolutionary dynamics. You will see a proliferation and diversification rapidly on these timescales. Yeah. So you see here the same kind of experiment, except now, instead of waiting for the spontaneous mutation and selection to operate on this resistant type, what they did was introduce the resistant host, and now you see that it rapidly increases to a level that was similar to what I just showed you before, and the viruses continue to oscillate. And the reason they're going up and down, especially the up part, which requires explanation, is that they are infecting and lysing and reproducing on the susceptible population, but the susceptible population is a subpopulation, if you notice these log scales, so that these oscillations are going to look like noise if I add those two together, and all I'll see is essentially a constant level of resistant hosts. So this phenomenon is termed cryptic dynamics. Because if you were to have observed, a lot of people try to look at time series and figure out interactions from patterns and correlations in time series. You have something constant, something oscillating, there's no relationship between them, right? So this notion that the interaction, the actual interaction is somewhat hidden or it's cryptic, in part because there's really subpopulation variation. Our level of measuring the population has obscured the fact there are really two variants, right? And one is susceptible and one is resistant. Okay. So there is one mathematical checkpoint here, so I just want to do this part briefly, which is, I haven't written down any models here, you can imagine them in your mind, but I want to ask this question, do resistant hosts always invade? Because I think this, once I've made this explanation, it should raise a question in your minds, why is the susceptible cell hanging out? Okay, so let's try for a moment to imagine we have a simplified model where we have these susceptible cells, the original cells, viruses. I'm going to skip the infected class. I've showed you before we can sort of eliminate it anyway and make this quasi-static approximation. The susceptible cells are going to reproduce. There's going to be some decay of both, right? And yet we can also have a resistant kind. So there can be a mutation event. And these also can reproduce, and they can also decay. But they are not infected by viruses. Okay? So we can imagine writing down these two parts. I think I've shown you that before, and I'll just write it down for completeness. And now I'm going to augment it with a new equation. I'll explain in a moment. Okay, so we have, as before, I introduced a similar notation. I've made a few little changes here where we have logistic growth in the absence of viruses. There's infection, lysis, and here I've made this rescaling where I've assumed the I is very fast. So I did this the other day. And we have loss of all of these kinds. Here I've made them the same. Here I've made it different. It doesn't really matter. The things I want to emphasize here is that there may be a different growth rate of this resistant mutant, right? And the total growth is going to be limited by the total, whether it's susceptible or resistant. Okay? So we want to ask the question, focusing particularly on this kind. And we want to think about the environment. And you see this as oscillating. And so this is the part where I'm going to invoke a steady state. You can tell it's not a steady state, right? Irrespective of that, we can think about the long-term average. We should average over a whole cycle. But for simplicity here, I just want to imagine that we're in a state in which we have some non-zero value of our susceptible host. And in this model, it should be, as you see there, limited by the viruses and not by the resources. And we have no resistant cells, and we have viruses. So that is our equilibrium. Here it's a fluctuating steady state. And now we're going to add a small number, or maybe if we were doing an individual-based model and we were asking the questions that Luca was asking, we just add a single resistant type, and I will make that connection in a moment too. So we're going to add a small number and ask, will it invade? So in this population framework, the question is, if I rewrite it, is this Malthusian growth rate in sense of an invasion growth rate going to be positive or negative, right? And so in essence, is there going to be more births than there are deaths? The birth rate, though, is going to be less than its maximum because there are already hosts there. Now, the hosts are less than K, so this is positive, but it's not inevitable, right? And so we can see here that invasion will happen when R prime 1 minus n star over k is greater than D, the death rate. So the effective growth rate exceeds the death rate. And I want to point out here, I've eliminated the n prime because there's so few of them, they don't see their own. We can ignore the fact that they're a very small number, so that's not the influence. The influence is the density of bacteria set by the resident viruses. Yes?
So you have the mutation here,
Yeah.
and you didn't include them.
If I'm assuming that I'm starting the model already with this n prime. If I were to write a full model, I would have to have a plus mu times here. I'd have to do all that. So I'm just assuming we've gotten to the place we have a three-dimensional system, what happens? Right? So when we actually do these sort of simulations, you're right, I have to discount very slightly the growth rate because some of them become the mutants. I have to worry about back mutation. But at the end of the day, what I really need to know is what is the fate of these mutants in an environment in which it's set by the resident susceptible cell and virus. Yeah, good question.
Can you erase the second n star in the last equation?
Yeah, this one is an m prime star, which now went to 0. Yep, right. And so we can also think that if r prime is some epsilon r, right, and if epsilon is less than 1, we can essentially write this as epsilon greater than d over r 1 minus n star over k. Which then finally I want to just plot this and give an interpretation. There's a funny point over here, but it's going to go like this. This x-axis denotes the extent to which viruses are effective at decreasing the host population. So how efficient are they at exploiting hosts? You can think of the limit. When n star is near 0, 1 minus n star over k is near 1. So this is an axis of increasing selective pressure by viruses. The closer we get to 1, the more it says that viruses are a major force of mortality, reducing the densities. This axis is the cost of resistance. This is invasion, and this is no invasion. So the point that I want to take you to take away here is that as viruses become a stronger agent of mortality, it is possible for a resistant mutant to invade even as the cost gets larger, right? So the cost of resistance is increasing as we go down because the effect on growth rate is substantial. Whereas when viruses have little to no effect on n star relative to k, then you have to have very modest costs. Otherwise, it will not be able to invade because a large cost won't make up for the fact of getting the benefits of not dying. You're paying so many costs by not growing as fast. Okay? So the point I want to make there, and the reason why I wanted to bring this up and not just proceed, is like my other, the first set of models in which you have a virus-host pair, the virus can kill the host, that doesn't immediately translate into a realization that the virus can invade a population and persist. In that same way, there can often be costs of mutations, or costs of resistance, and if those costs are too great, depending on the ecological context, that can lead to invasion or not. So just because there was a resistant host doesn't necessarily mean it's going to invade. And likewise, I should point out, and I'm not going to go through all the analysis, that when it does invade, it doesn't necessarily mean it displaces the susceptible host. Because there is a cost, and what you can see, you can imagine happen, is that because of these fluctuations, when there's a lot of viruses around, being resistant is more beneficial. But as the viruses start to drop, now neither the susceptible nor the resistant host is paying the cost of the viral mortality, and so the susceptible host can grow faster. So it leads to coexistence between all three kinds. Okay? So the point, now we have something in my screen that's doing something. Right. So the point of the first lecture was really to introduce the notion of viruses and hosts in terms of this predator-prey analogy, and the consequences are these canonical predator-prey dynamics. And most of this, frankly, has been known since the late 1970s by Bruce Levin, Chow, Stewart, and others. And in the 90s, there were these empirical examples with initial models to explain how when one, or but not both, whether the viruses or the bacteria are evolving at the same time scale as the population dynamics, then you can get new kinds of phenomena when viewed at the ecological scale. And again, here what I mean by evolution is a heritable change in the frequency of genotypes in a population. So we have a population that has now two subtypes. We're looking at the total, and we see a difference when we look at the total population dynamics. And this can be cryptic cycles, but I also want to point out, or these things, something a little bit more general. So I just gave you an example of cryptic cycles. It's one example, so you might wonder, how general is this phenomena? Is this something that I've just cooked up to show you for these lectures because that's convenient? There's a nice paper in 2014 by Hiltunen et al. Working with Steve Elner and Hairston and others, in which they re-examined population dynamics literature and asked, do these predator-prey time series look like canonical Locke-Volterra-like dynamics? Or when you plot them in the phase plane, instead of going counterclockwise, do they do things like either have cryptic dynamics or even in-phase dynamics, where they're essentially antiphase, where the peaks and troughs are coincident rather than being shifted? And what they found, this is one example, and now we're moving away from viruses for a moment, where the bacteria now are the prey, but amoeba are the predators. And you see that the peak and the trough coincide. Right? And here's a model in which that happens. And the reason why this can happen is that there were these defended strains of bacteria, fast-growing bacteria, and the relative frequencies of the two genotypes are changing at the same time scale as population dynamics. And what they found in reviewing these apparently canonical predator-prey examples, about half of the examples didn't actually look like Locke-Volterra. So I think that's another important point to take away, that we have this very dominant idea of Locke-Volterra-like dynamics. But as soon as you start to realize that evolution can be fast, meaning strains or subtypes are changing in frequency at the same time scale as population change, then very new kinds of phenomena, qualitative phenomena, can arise. Okay, so we have counterclockwise, we have in phase or cryptic, right, going this way or this way, if you can imagine the plane. And then the last one that I want to talk about in this component are these multi-type predator-prey dynamics, or coevolutionary dynamics, in which instead of having counterclockwise predator-prey oscillations, we can actually run the system in reverse and get clockwise dynamics. So let me explain how that can happen. Okay, so I'll go back to the population dynamics of viruses host example, this time with multiple strains. So first it was one virus, one host, then it was two hosts, one virus. Now we have two hosts and two viruses. And I'm not going to belabor the point here, but you can trust me that we write down these general sets of equations, which look like the same as before. We have two prey, two predator, they interact, there's growth of the prey, and consumption or predation by the predator or viral class. And this is just to give you an output of what the model looks like, where the total is solid and the strain level is in the dashed lines. And there's a lot going on. The most important thing I want to show you is that you see this predator or virus peak precedes the host peak, as if the hosts are eating the predator, right? In fact, there had been a mislabeled time series from back in the 70s which had this one. It turned out to be mislabeled, which raises the question, do hare eat lynx? No, lynx eat hare, right? So the phenomena here, just in this one example, shows you already that once you have multiple types, it's possible for there to be an offset where right before the host peak is the viral peak instead of the other way around. So why does this happen, and how and when can it happen? So let me now move back into this phase plane where we have total prey and total predators, just reminding you that the usual way for predator-prey oscillations to run are counterclockwise. And this is an example in which they're running clockwise. And to explain and understand how this can happen, let me highlight 4 points on this cycle. Each one of these 4 points keep in mind there are 2 genotypes of each kind. There are 2 viruses and there are 2 bacteria. What I've done here is label the relative frequency of the prey in blue and the predators, the virus, in red. So that you can see that when we have a lot of solid, that means there's a lot of low offense viruses, and when there's a lot of open space, like here, these are more virulent, able to infect or lyse more rapidly or efficiently types of viruses. Likewise, for the blue, when we see this dominant blue color, that means there's low vulnerability, dominated by the low vulnerability type, so the good at defense type, and here it's high vulnerability, better growing type. Okay? So let's start over here. If we start in the right-hand corner, we have a lot of prey, right, and not that many predators, and we're being dominated by the low vulnerability type. Right? But we don't have that many predators around, so somehow, by not being very vulnerable, there's a lot of prey. But because there aren't many viruses around, the system switches. Those genotypes that are highly vulnerable can start to increase in relative abundance. But as they increase in relative abundance, then the high offense virus will do better as the system is dominated by low defense prey. So we see this switch happening to the point where now the system has quite a lot of high vulnerability prey, right? But the numbers have actually been driven down, and the predators are doing well, except now that these are highly vulnerable, there's a switch from high offense to low, because why pay the cost of being this very offensive type when everything is vulnerable to you? But now when the system is dominated by low offense predators, then there can be this other switch back, because being a good defensive prey will keep you around. So not only do they increase in relative abundance, but the total prey increases. Right? And then you see the switch again to low offense, there's no point in paying the cost, to now a high offense kind, because the only way to overcome this low vulnerability is to actually have the high offense kind. So we have an oscillation here in the genotype space that's concurrent with changes in predator-prey population dynamics. Right? So this clockwise population dynamics is driven by change in the genotype frequencies. Okay. So this seems, although interesting, and we can find this sort of robust result for this kind of scenario where we have 2 kinds with high offense, low offense, and a highly vulnerable, low vulnerability that trade off with growth. Do we actually see this in data? So let me get into that. So are there clockwise cycles in phage bacteria data sets? So I'll go back to the chemostat example just to remind everyone again, this is a picture of an actual chemostat. This is continuous flow. You're flowing in nutrients. Everything is flowing out of the system. And this is a study from Bruce Levin's lab where they were looking at interactions between Vibrio cholera and its bacteriophage. So Vibrio cholera, which itself has a pathogen, has phage that infect it. There's an interesting backstory there. But for the purposes here, these are the resulting population dynamics. Again, you see large oscillations, viruses and bacteria. This is a 600-hour time series. And the way this was described was that they were complicated population dynamics. Right? And so I want to highlight 2 components, which I've bolded. And I'm going to take these 2 subsets of the time series and move them into the phase plane. So these are from time 48 to 119 hours, 119 to 218, and then these 2 other subsequences, all of which seem to wrap around in the clockwise direction rather than in the counterclockwise direction. And I want to point out here that this also seems in accord with the mechanism that was going on in this particular experiment. So they started with a single pair, but what they found were there are actually 2 kinds of viruses. They call them phage T and B. T is turbid plaques. It didn't seem very efficient at killing hosts. And B were big plaques, which were very efficient at killing hosts when you actually do these plaque assays. And there are also 2 kinds of resistance. One which was negative to the T, and one which was negative both to T and B. So therefore, you had a continuum also of the resistance. So we seem to have the right ingredients here, as well as a potential explanation for how counterclockwise cycles, which are expected from predator-prey dynamics, can go in reverse when there's concurrent change in genotype frequencies. Okay. I also want to point out, I mean, if you look here, these are very, very short time series. So one of the concerns I had, Mike Cortez is a mathematical biologist. My background is in physics and now quantitative biosciences, ecology. I was worried that these were too short to really get decent statistics on. And so one of the things that we tried to do was then generate an ensemble of artificial time series in which the point-to-point variation was the same, but we then randomized the occurrence. So we have time series of phage and bacteria which point-to-point variation and the overall statistics are the same, but we've messed up all the correlations. Because it's possible by chance we take 4 steps and we make a loop. And the way that we then evaluated this were 2 ways, one of which we calculate the winding angle and ask, do you wind 2 pi around the circle, right, for clockwise? And also, how far are you in terms of this phage bacteria densities? Do you come back to where you began? So do you essentially go like this and come back to where you started? And so if you do, this would be a paradigmatic where the diamond is clockwise cycle. These are the observations in these 4. There's these cloud of points, and it ends up being statistically significant, which then if you contrast, this is the lynx hair example, which is accepted in the field as counterclockwise. We did the same thing, and you can see we get essentially the similar statistics. So at least we have confidence here that to the extent to which people accept the lynx hair example as being an example of counterclockwise cycles, that these phage bacteria data sets look like clockwise cycles. And I'll generalize it a little bit more, that we then went on to find a number of other examples as well. So these are all examples in which you have these sort of clockwise orbits rather than counterclockwise orbits in the phase plane, spanning quite a number of systems, phage, bacteria, mink, muskrat, and so on. Okay, so this generalizes finding of reverse cycles, and I want to just get these ideas in your mind in terms of a single synopsis, which is that when there's no evolution, we expect from basic predator-prey dynamic theory that we should expect counterclockwise cycles. When there's either predator or prey evolution, you can get new dynamics like anti-phase cycles or cryptic dynamics. I want to point out you can also get normal counterclockwise cycles, but you can't get these sort of dynamics without appealing to this subtype variation. And also this should be evident for anyone who's done nonlinear dynamics work, is that if you only had a 2-dimensional system, you can't be at one space in the phase plane and move 2 different directions. Right? So that suggests you're missing some component. You've been projecting a higher dimensional system onto a lower dimensional system. And finally, when you have coevolution, then you can get these clockwise cycles, and you can also get these anti-phase cryptic dynamics as well as traditional cycles. Okay, so the summary here is that rapid changes in the frequency of genotypes can have effects on ecological dynamics, so there can be an interaction between ecology and evolution when those things happen on concurrent or similar timescales. We often make the assumption that evolution is slow and ecology is fast. And I'd hope that you can come away from this part saying, no, that especially when you're dealing with virus and bacteria, but this also applies to Daphne and rotifers, applies to other systems, it's certainly experimentally tractable within virus and bacteria, but changes in the relative frequency of genotypes can occur and does occur on similar timescales to changes in total population dynamics. Okay. This is nice, but what other dynamics occur in more complex communities? And to go in that direction, so we have 1-1, 2-1, 2-2. It would be a very long and boring lecture if I then did 3-2 and 3-3, et cetera. It would take forever. So what I want to do instead of going in that direction is to change gears a little bit and go more in the empirical direction to get more traction on the kinds of networks that are seen in nature and use those as a guide for beginning to understand what kind of dynamical processes might underlie their coexistence. So in this last part of this first set of lectures, I'm going to try to answer this question: what is the relationship between infection networks and host viral dynamics in complex communities? And this is work spanning 3 different PhDs, one by Cesar Flores, the second by Luis Jover, and the third ongoing by Ashley Conan. So I want to step back to what I mentioned already in my first lecture, is that viruses are quite abundant in natural systems. And I gave this example from 1989 of one of these early papers that found 250 million viruses or virus-like particles per milliliter, a very large number. And again, you might also be skeptical. To what extent was this an exception? This was one example from 1989. What's the update? So we were also curious for a number of reasons And so I co-organized a working group trying to get into understanding ocean viral dynamics. And what we did is went back to the literature. Any space you see a point was an open ocean assay, so it's a group of oceanographers going out into the environment, measuring both microbial abundances and viral abundances. There's greater than 5,000 samples spanning 20 plus years, quite a lot of diversity in terms of seasonality, and also looking at the surface and at depth. So when we compiled this data, these are the prokaryotes. You can think largely bacteria, viruses. This is log scale. This is a 1-to-1 line and a 100-to-1 line. There were also some dominant ideas that this data should fit nicely on a 10-to-1 line. I think you can see it does not for all sorts of reasons, and I can explain that later. What is, in fact, the reality is that there's quite a lot of variation. 2 orders of magnitude variation that's often typical in terms of the ratio of viruses to bacteria in natural systems. But there is a strong tendency for the viruses to be more abundant than bacteria, and again, emphasizing the actual values here, right? This is 10 million, 100 million per milliliter. So this finding of hundreds of millions per milliliter was a little bit on the high side in terms of the distribution, but it's certainly not uncharacteristic in the data set. So these are incredibly abundant in natural systems. And that's interesting and important because we know that virus-host interactions modify sulfate. That in turn modifies populations. And if we multiply that by these enormous numbers and enormous interactions, this is another reason why viral ecologists want to figure out what are viruses' role as ecosystem modulators, not just as agents of mortality at the cell scale or modulators of population dynamics, but what roles do they have in ecosystems? And so one of the most important ones they potentially have is to lyse cells, redirecting cellular components back into the microbial loop. This is called the viral shunt, and you can see it depicted here, where instead of this heterotroph or this autotroph being grazed by a grazer, which gets eaten by some other zooplankton and then goes up the food chain and eventually becomes fish, it remains small, keeps essentially organic matter small, which can be remineralized by heterotrophs, and again, staying in this microbial loop. It also has interesting consequences for climate, for CO2 fixation, export, and so on. And so these are heterotrophs and autotrophs, but there are a lot of heterotrophs and a lot of different autotrophs. So yes, we know there are a lot of viruses, but not all those viruses are infecting all the bacteria. And yeah, we can break it down at this level, but we need to go even further. So one way to go further is to really characterize the diversity in a community and begin to have a census of who's there. And if you know who's there, maybe you could figure out who infects whom. The challenge with that, this is from work from 2005. The situation hasn't improved that greatly, which is, this is the number of viral genotypes, and I'll just give you the quote. This is from a marine sediment sample where they were trying to estimate, similar to species abundance relationships, but for viruses where species definitions are a little bit messier, so there's an effective definition, but 10,000 to 1 million viral genotypes. That's the range. So first of all, you should take away a lot of different kinds. It's not just like there's many viruses, they're all of the same kind. There are many viruses of many kinds. The second point you should take away is that viral diversity estimation is not very effective or robust at the moment. If you want to learn more about how to deal with estimation problems and why estimating the total number of species or genotypes in a community is hard because it relies on estimating a tail, and the tail is what you don't see, you can take a look at this paper, Bart Hageman et al. And Ismi J. There are other features of richness and diversity that we can estimate, like the Shannon diversity or Simpson diversity, but estimating the total number of species from a subsample is inevitably going to be quite difficult if you decide you want to do that. So we have a lot of different kinds in this environment. So rather than, as I said before, going 1-1, 2-1, 2-2, etc., we decided to take a different tack. We wanted to know, were there any characteristic patterns of who infects whom in natural environments? And so how do microbiologists and virologists do it? What they do is take some set of bacteria and phage from an environment and then use what are called spot assays or plaque assays to determine who, in fact, infects whom. And this is what one of those looks like. This is an agar plate, bacteria growing happily. So to grow bacteria, you give them food, resources, carbon and other media. To make viruses replicate, you need to give them bacteria. Right? So first you have a lawn of bacteria, and on top of them you put your virus. These are the viruses. This is the bacteria. You'll notice the viruses spread and make a clearing, or essentially what is called a plaque, on this agar plate. So here, this virus can infect this bacteria. Sorry, there we go. And this virus cannot. You don't see any clearing. So they try, and then they see that it fails, and that's a 0, and this is a 1. And people do this again and again and get these sorts of tables, which we digitize, and we get these matrices, these phage bacteria infection networks, and I will be using a convention where phage virus and bacteria are on the columns, and the rows are hosts, and the white squares denote the fact that that particular phage can infect that particular bacteria, and the blue background means that they tried and it can't. Right? So it's not the absence of data, but actually they tried and found that that was a 0. Okay. So this is one example, and we then collected over the course of the summer as much data as we could find at the time as possible. These are some of the images that you see, the 38 studies, greater than 12,000 different infections, quite diverse in terms of whether they come from natural environments, from eco-evolutionary experiments, right, or from lab strains. And it was about an equal mix of sort of ecology and evolution. We had hoped at the outset that we were going to see a characteristic pattern there that was going to inform us about the kind of models or systems that we would want to explore and explain. And I don't know about you, but when we looked at it the first time, it just looked like a mess, right? There's a whole bunch of different ways to look at these things, and then that actually became the insight. So if you can imagine, so each one, and I'll explain what those are. In many cases, they are strains of the same species. Sometimes they are strains even at different genus levels, but that's an important question. I'll get back to it in a few slides. Yep.
By definition, you can only do this with bacteria that you can culture.
Another thing that I will get into later. Yeah.
I keep hearing about these ocean studies. 99% of the bacteria can be cultured.
Correct. So both those questions represent, in essence, the next 2 modules of this talk. So I'll address both. Yep.
Do you rank order?
Say again?
Do you rank order?
Right, so how are these ordered? And that's the question I want to answer right now. So you can imagine an experiment in which a group looked at 20 bacteria and 20 phage. How many ways are there to represent these matrices? Well, I can choose which bacteria to put in the first row and which in the second. I have 20 choices, then 19. So I have 20 factorial times 20 factorial. It's a very large number of ways. And so I'm just showing you here the ways in which they looked at them. And the ways we looked at them are as follows, which I'll show you in red, highlight those that have a characteristic feature which we call nestedness. And by nestedness, and I know Paul is not in the audience right now, but I'm going to evoke his name, there's a notion of matryoshka dolls where one is nested inside the other. Here we have a notion of nestedness. I'll show you some of the best examples where here are all the viruses that this bacteria is susceptible to. The next one is susceptible to a subset, more or less, and so on and so on and so on. Likewise, here we have a virus kind that can infect almost everything. And then we have less generalist types, but that are infecting not different sets, but a subset of that generalist virus. And in fact, if you get all the way here to the end, the specialist viruses are specializing on hosts that everyone else can infect. They're not specializing on hard to infect hosts, they're specializing on easy to infect hosts, which also raises certain questions. Okay, so just to give you an example of what it is we did, here's this original data. This is the same data set where the rows and columns are rearranged. Right? And so this goes back to your rank ordering. It's not quite just a rank ordering in terms of the marginals, but that actually is not such a bad thing to do. Right? So these are designed to find that ordering which maximizes what we call this nestedness, the degree to which there are subsets in terms of infection ranges. Okay. And I just want to make one more point about this. I think for the most part, you haven't heard about it, so maybe this is unexpected to you, but was it unexpected in the literature? And for the answer, how do we actually evaluate that? Did people know this or not? And so whether or not they knew it in their minds or not, we tried to ask what was the nestedness of their sorting and their presentation versus the one we found. And so the answer to some extent is yes, that we have these nestedness in the original sorting, which you can't see anything, and they pop up to some of them are nearly perfectly nested had they actually arranged or rearranged these columns and rows. So this is important for a number of levels. First of all, they're very different systems. And so there's a temptation to think each one is different with its own unique set of rules. And to some extent, that's always the tension between physics, mathematics, and biology and ecology. The point we're trying to make here is that it's possible to at least think about these infection networks in a common framework, and maybe there are common organizing principles that we may be able to apply across different systems. The other thing, just from an empirical side, even if you don't like modeling, the fact is that we found a continuum from generalists to specialists in terms of viruses, where again, the viruses tend to specialize on the easiest to infect hosts. And that is something that is becoming increasingly found, and people have seen it multiple times, particularly when we do ecoevolutionary experiments. It's a feature that seems to arise again and again. And if you want to learn more, there's a review of this and some other models. I'll give you some more references later. Okay, so that's the empirical side. And then we decided, now that we have some sense of what a community-level infection network might look like, we could at least begin to ask the question, how do they coexist? Or how could a set of viruses, which each don't have their own niche, they're overlapping niches to some extent. A virus doesn't have one host, and another virus has another host. They're all sharing hosts. So why are they able to persist, particularly with the following dilemma? Here's a stylized version of one of these nested networks, and I want to ask the question, why doesn't the most resistant host outcompete the rest? Right? So it's susceptible to virus number 5, right? But so are all the other hosts, and they're susceptible to a whole bunch of other viruses too. And likewise, why isn't the most specialized virus outcompeted by the rest? It can only infect one host, the easiest to infect host that everyone else infects. All the other viruses get benefits from infecting all these other hosts. Okay, so you may be able to anticipate one potential answer. What we did was take a similar framework to what you see right here on the board, where we have hosts and viruses. We now have strain types I and J, and we have competition logistical among them, lysis, virus release, and decay. But importantly, there's this capital M, which denotes who infects whom. So these are these networks that we just spent a lot of time analyzing. And until that point, the dominant idea had been these kill the winner models, that there really was one virus per one host, and each one particular virus controlled that particular host density. So it's similar in spirit to these models, except now we're going to assume that cross-infection is structured, and there are these overlapping ranges. Okay. So I'll give you one short example of what might happen in these systems. What am I doing on time? I've totally lost track of time. When did I even begin? Sorry to interrupt my own train of thought here. 11:20, okay. Oh my. Okay, that's fine. I'm somewhere in this talk. Okay, so we take now a 2x2 matrix, which has, you can imagine, perfectly nested, so we've just taken out the bottom right corner, and I'm going to have two simulation results here. It's the same network, both have 2 viruses and 2 hosts with 3 interactions, so there's one missing. And in one simulation, only 1 virus and 1 host coexist, and in the other simulation, all 4 coexist. So what's the difference here? Somewhat rhetorical question, but I'll take a thought. So we have this simulation of these community-level models of virus-host interaction networks, same community, same set of rules, but we have 2 different outcomes. Someone I haven't heard from who's willing to just help me move along, or I'll just move along myself. Anyone have a guess or a stab? Resistance? Okay, in this case, that could certainly happen, but I'm not going to allow mutation in the model. And it's also not an initial, because we're focused on ecology, it's not a difference in initial conditions. The difference is that I have to choose life history traits. So it's good that I have these on the board. Every time I do this kind of model, I have to have a different adsorption rate, maybe a different effectiveness of lysis. So I have the same network in terms of 1s and 0s, but these interaction networks are quantitative. And what I've done here is made an informed choice about what life history traits the viruses and the bacteria have. And what we found is that if you look at this 5x5 example, but it can be scaled up to arbitrary sizes, that if there's a trade-off so that the most vulnerable type grows the fastest, so in other words, an extension of this idea to the community scale, and the least vulnerable type, the most defended type, grows the slowest, and likewise, if the specialist can draw down the host that infects to a lower level, right, so it's this idea, whereas the viruses that infect many things don't draw them down as efficiently. So if we have trade-offs of life history traits and range, what we can see, both prove mathematically, but also show from these simulations, is that you can get arbitrarily large infection networks where all the players coexist, even though none of them is infecting a unique type. Okay. So that also raises the question, is there evidence for any of these trade-offs? Right, so trade-offs seem to be quite important here in driving coexistence. And so I want to say one caveat. It's certainly true that there are examples, very strong examples, of trade-offs between growth rate and resistance. You take a susceptible type, there's a resistant mutant, and now it suffers a growth rate cost. These are some examples, not the typical ones. Viruses are T4-like potoviruses, and this is a cyanobacterial host, so these are very important ecology players in the marine environment. This is the wild type. These are all resistant types. Here they have a growth rate cost, but you notice there are other examples in which you can get resistance, in essence, for free with respect to growth rate. Likewise, this is a different example of fitness versus host range. These are all mutants that enable a host range expansion of an RNA bacteriophage 5/6. And in each one of these cases, the fitness on the original host goes down, except there are 2 cases when it doesn't. So I guess the point I want to leave you with here is that this notion of trade-offs, I think, is a tendency, but it's not a strict law, right? And in fact, in the case here, Debbie Lindell and her group were puzzled by it, and they found that often when you have resistance to one phage, you end up becoming susceptible to another, so this may end up being more like whack-a-mole rather than a direct relationship between growth rate and resistance. Okay. So certainly there's a relationship between trade-offs, so we seem to be on the right path. The other thing that I think is questionable about the model is that we've assumed perfect nestedness, whereas you saw all the examples were not quite perfect nestedness. So what we then did is ask the question, if we have trade-offs and the networks are not necessarily perfectly nested, but go across a continuum, is it true that being more nested would tend to lead to increases in biodiversity, or the number of strains locally in this environment? And so we found the answer to that was yes, by essentially reorganizing, rewiring these networks with trade-offs and finding that as the network became more nested, then there were in fact more types. And this applies to a whole bunch of different cases, though this has to do with whether the trade-offs are strict. So we began to explore the tendency for nestedness and trade-offs to together combine to lead to more types. And we found the answer is yes. So in other words, nestedness can promote diversity, but it need not be perfect. Likewise, trade-offs promote diversity. They don't have to be perfect. And that's really the major takeaway points. Viruses and microbes are incredibly abundant. They're diverse in natural environments, and also relatively specific in whom they infect, but not so specific that they only infect one type. They can exhibit this range between generalists and specialists. And yet, despite the fact that they don't each infect their own type, we see coexistence is possible. And it's supposed to do the next 2 parts of this talk in 5 minutes. So I'll maybe do one part and just mention the other, and I'll skip to the end. I want to go back to the question that was raised. What was the actual, let's say, phylogenetic diversity of the kinds that we analyzed? So I think you asked this question, Nathan. And so here are all the different types of host strains that were seen in this 2011 analysis. And the arrows denote the fact that there can be, for the most part, viruses within, viruses of hosts within a particular species. But sometimes, very rarely, we see viruses that can infect strains between different species or even different genus. Okay? And so one of the things that we worried about was that our finding of nestedness, this conclusion that this is a characteristic feature of natural networks, was biased by the fact that these are culture-based. And if you've ever tried to work in a laboratory, you will find that the cyanobacteria laboratory and phage are not the same as the E. coli bacteria laboratory and phage. You're not regularly co-culturing those 2 diverse organisms and then trying to see if there's any cross-infection. So you can imagine the thought experiment. Here's a almost perfectly nested network. Here's another one. This is E. coli and phage. This is a flavibacterium, a fish pathogen, and its phage. Each are nested on their own. If you had done the cross-infection, do you really expect that many infections? Probably not, in which case you expect probably modularity. At this very large scale, we might expect modules, and inside modules, we might expect there to be some nested structure. Now again, we're a theory group, we collaborate with empiricists, so it wasn't easy for us to test this idea directly, So instead, we went back to the literature and found one example in which we thought the scale of the study might permit at least some level of test of this idea. This is a paper by Carl Mobus and Nat Kemper. It's from 1981. It's a journal that I'm sure you're all reading, the Helgel Mira Sunters. It's quite an interesting study, well ahead of its time. They've been a bit forgotten. I mean, I know this literature fairly well, and I was not aware that inside this paper was a fold-out table. Literally, you had to fold out like this, and it ends up being about this big when you take it out of the journal. And inside are these dots. And each one of these dots represents an effort by Mobus and Nat Kemper to cross-infect a particular isolated bacteria with a phage. Hundreds and hundreds of times. In fact, if you look at this, I'll show you in a moment, these were collected from 48 stations of the Atlantic Ocean. We digitized this, which itself was its own challenge. But there are 286 bacteria, 215 phage. They did this more than 60,000 times. There are very few, even now, very few studies of this magnitude. There are now a few studies of this magnitude. And for the most part, the answer was no, which also tells you something about science and the effort involved. So there's a lot of futility here, but there were a number of about 1,300 positive interactions, and they noted that it looked like some complex, right, or complicated type of interaction relationship. And I saw a question up there.
Were they in the same environment?
No, so they're all marine environments, but they were taken, each one of these red circles, there was a cruise across the Atlantic Ocean, and they sampled phage and bacteria from each one of those environments. And
Like in the same, like,
in the water, right, I'm trying to remember if it was, I think it was near surface water. I'd have to go back and look.
yeah,
Yeah.
because I don't know. Maybe the modules are like
I'll show you some more in a moment, right? But that is definitely true that if you look in the surface or deep, but I'm almost certain that these were taken relatively at the same location near surface waters. I will have to double check, right? But it's a very good point. Yeah, they could have done a different design where you just go at depth, and you might have seen the same thing too. But they went to see. Okay, so you have 1,300 interactions, and this is what it looked like. And what we first tried to do was, well, is this nested? Ask the same question we were asking before, and I think you can see that it's not particularly nested. But instead, there are standard ways now to look to see if there are modules, and this is the same data organized into 49 different modules. And I hope you can see that there's almost no positive interaction points that don't sit on this block diagonal form. And there are different ways to do these modularity algorithms. We're using a standard bipartite module because we have phage bacteria rather than a unipartite modularity algorithm. So this is incredibly modular in the sense that there are groups of phage that preferentially infect groups of bacteria and not others, right? And likewise, groups of bacteria that are preferentially infected by a group of phage and not the others. Yes?
Can't you play the game backwards? You ask the computer to arrange it in a way, then find out what's the axis.
So here, in essence, what we've done here is find the modules, and we've just organized them by size. And so the display is simply that the modules have to do with whether or not if two of us infect the same bacteria, do we tend to infect, do we share a triangle, in essence? But once you decide that, then there is some question about how do I order within, which I'll do in a moment. But then this establishes we're just organizing them by size. Right? But you can't order it by the row or column sums as I did before. That would be a nested arrangement, and it doesn't look like that at all. Yeah, so I just wanted to contrast again. This is the original. This is their specialization in cross-infectivity when we look at these large-scale, at least environmental studies. Yes?
Now you seem to have that specialist infect just one bacteria. So is this because this belongs to different cultures?
Yeah, so it's not just one, so let's go in. Remember, there are 215. These blocks are not one. Inside them are dots. I hope that everyone sees that. There are 215 columns here and 286 rows, and maybe it'll be more evident by me zooming in and showing you that they don't just infect one thing. The blacker bacteria, whiter phage, you see they can infect more than one thing. This is just an example of a module, so there still remains cross-infection inside the modules. Okay? And just to point out that these modules are also comprised preferentially of local geographic regimes. So there is a sense that the locality mattered, but I also want to point out that they were able to find a phage from thousands of kilometers away that can infect a bacteria from the future. Okay, because they were collected at different times. It takes them a while to sample. So that also tells you something about the way in which, when we look in these environments, there is some notion either of stability or functional redundancy that things are able to affect from a very different part of the ocean because of other similarities. And so just to answer the question I think that you were raising, when we look inside, we actually still find, though they're not as clean, this notion some of, statistically speaking, generalists and specialists, even within the modules. So we have at the large system scale, we have modularity, but inside the modules, we may see again nestedness. So the take-home message here, and I think this is more or less, I will maybe just do 2 plugs, but I won't do the other bonus part of this talk, which is that, again, nestedness is a hallmark of many systems, particularly when we're looking at microevolutionary or small system scales. Trade-offs can help facilitate coexistence, but that's not the only structure, so I don't want you leaving here today thinking, well, all these phage bacteria are nested. No, at large scales, we expect to see a different kind of structure, which we saw here, which was modularity, but there still be even a hierarchical structure. But it's clear then that we don't yet totally understand these phage bacteria networks and their consequences at community scales. So I'm not going to have time to go through this last portion. I mean, if people ask questions, I can go revisit, but I just want to make a plug for it, which is this question of who infects whom, irrespective of culture. So irrespective of whether or not you can isolate that virus and bacteria in the lab, has become one of the most important parts of the technological or molecular biology side of viral ecology. So this is one example of an effort led by Matt Sullivan, a collaborator who developed a method called viral tagging, in which you essentially fluorescently stain or tag the viruses. And then you have a bait host, which you've cultured using radio-labeled isotopes, so that you can then mix them together, put this through a flow cytometer, look for the bright signal, and then essentially separate the heavy from the light DNA so that you can figure out which viral types got into that bacteria. Sequence the viruses and say, who were they? And this is what you're looking at. Okay? What you see are all these different groups. Many of these had never seen before in nature. We thought we understood who the major viruses were of this particular dominant cyanobacteria, but it turns out there were a few examples which we knew, but there was a massive culture bias. And there were all these different phage, just like there's a culture bias for bacteria, we also had a culture bias for the viruses. Massive numbers of dominant phage, which we had never seen before. This is not the only method. Other methods, digital PCR, amplified genomes, polonies, etc. So what is a modeler or a theoretician to do in this space where it seems like technology is driving the ship here? There's a big caveat. Almost all of these are focusing on a particular group where you know something about that group. So in this case, you have to have a bait host, which has to be cultured, and you're using a culture-based host to pull out unknown viruses. And actually the method is also quite complicated and tricky, which applies to most of these as well. So what we have wanted to do for a while, and I'm just going to plug it, and I can happy to talk about it with folks over breaks, is to keep in mind that we don't know these weighted interaction networks. We don't know who infects whom, but what we can often observe through direct sequences is how abundant are the different kinds. So it's possible through both viral metagenomics and cellular metagenomics to keep track of how many different kinds or the abundance or relative abundance of types in an environment. And presumably, if viruses are shaping populations, if we knew that, we could do this forward problem, which is what I've been describing. I prescribe the network and say, given that network, what are the types of signals we see? But increasingly, we're interested in the inverse problem. If we can measure this, could we infer that and tell you at the community scale without sequencing them who interacted with who and even what were the quantitative rates? And I just want to point out there are 2 broad approaches to this, correlation-based and model-based. And I'm not going to go into the details because I'll just show you just one picture at the end. But these correlation-based approaches, which have tended to be used in the literature, which say that correlations of these observed relationships are proxies for interactions, I'll just tell you that it's not the case. Where is my slide? Whoop, no, I'm going too fast, going the wrong way. The answer is it's not the case. We've done it, and I can tell you more about it. It's in M systems. It doesn't work for all these nested interaction networks and modular interaction networks and life history trait variation. You do not recapitulate this structure from things that look like this, despite the fact that they're often commonly used. I can't recommend them, particularly for these strong mortality kind of or antagonistic interactions. What we have been doing instead is something called model-based approaches. So we take these models, just like I've shown you before, and then we ask the question, let's in silico simulate the virus host dynamics, take measurements of the relative changes at these times, so this is why it's blocky. We'll call this the W matrix and the H matrix, and the abundance of hosts at a given time. And then by taking a look at the expected per capita change in viral densities, we expect it to have a contribution from these cross-infection networks, which are unknown, times the measured values of the densities, minus the unknown decay rates times 1. So each type is decaying, but it's being resupplied by lysis. So this sounds a bit of a mess, but really this is a regression problem or an optimization problem. We have these unknowns, we have measurements, and we'd like to find some unknowns that best fit the measurements. So that's what we've done. We've set this up as a convex optimization problem. The work of Zhao Xavier, who's done this experimentally and in silico for micro-microbe interaction networks, has taken a regression approach, the same principles. The idea is that you want to minimize these observed per capita changes minus the expectation, subject to certain constraints. And you can also impose other sorts of constraints. This is the original matrix. This is a reconstruction. It can get both structure as well as quantitative details, which gives us some level of optimism that it's at least worth trying. And that is what we're trying to do now. So I can't give you the results yet because it took us a while to convince reviewers that it would be worth actually doing these experiments. And unlike some indulgent experimentalists, we have to get a boat, research expedition. So to get this to happen, which was really what we want to see, can we do this in natural systems with ecologically relevant players? So the idea is that we're going to go to sea, our first expedition. It's led by Steve Wilhelm at the University of Tennessee, Knoxville. Myself and Matt Sullivan is going to go to natural systems, measure communities of viruses and hosts, try to do this infection network, and do it both from strain collections for which we know the answer, so we can actually look backwards to see how well we did, but also from natural environments for which we don't know the answer, and we'll then have to fill that in with some of these other techniques. So I'll tell you what happened in about 3 or 4 years, maybe a little sooner. Okay, so let me just wrap up here to say that the reason why we've gone through all this effort, both to try to think about what viruses might do to population dynamics, but also increasingly think about the reverse problem, is because of this role of viruses as ecosystem modulators, but we can't figure out the particular levels of carbon and nutrient regeneration or the relevance of virus to ecosystems if we continue to assume that it's one big black box where all the viruses interact with all the hosts. We would like to identify those pairs which disproportionately play a role in regenerating carbon and leading to mortality events of dominant microbes. And hopefully we'll get there in a few years with the help of colleagues. If you're interested, especially these first 2 lectures on the kinds of approaches, the models and so on, I wrote a book on the topic, Quantitative Viral Ecology. It was released in 2015, though it came into print really in 2016. The first chapter is available online at the Princeton University Press website. And there's also a bunch of other code and stuff associated with this, so if you're interested, feel free to reach out. And with that, I think I've taken more than enough of your time for today, and I'll be happy to take some questions. Thanks.
So we're not going to make a break before the questions, as I told you. We have the colloquy at 2 o'clock, just to remind you. So we are open for questions. And after the colloquy, you can make more questions if you want.
There's a second mic here if you want to. You pass it, at least just here. Yep, in the back.
I was curious how this trade-off works mechanically, because like I always thought that bacteria, their infection was caused by, if it cannot be infection by a virus, it's just like that protein that is on its membrane, it can change the protein, so it changes the virus that can infect it and has no energy costs, and you blew my mind. And
yeah, so what are some mechanisms? So first of all, if you have a change in moiety of let's say, a receptor that has something to do with nutrient uptake, if there were no cost, it would be because there's a large configuration space, protein space, which have the property that nutrients come in at the same rate and no viruses come in. It turns out there are examples in which that is not necessarily feasible within a one step from where they are now in sequence space. Maybe that other place exists and there can be compensatory mutations, but the initial mutations, either because they're not as efficient at taking up resources, other mutations could be a deletion of the receptor entirely, and then you can have a greater cost. There can be other mutations having to do with transport from the periplasm, which leads to other issues in the system. And keep in mind that some of these changes in surface are not just like the classic example, like the maltose uptake receptor, but they also may be LPS or other features which changing those, there are consequences to the overall physiology. So there's lots of examples from inefficiency in taking up nutrients, transport from periplasm, and other physiological effects. But as I pointed out, there can be examples, there can be compensatory mutations, and there also can be examples where they don't see a direct cause, where you're right, it shuts down something. It also, one last thought, depends on context. So growing in a maltose minimal media, if you mess with the maltose receptor, you're in trouble. If you're growing rich media, sure, knock it out, you might not have a fitness cost. Yep. Here on the side.
Hi. In the module diagram, are the group of bacteria and group of virus related in some way?
Yeah, so that was a question we wanted to ask, unfortunately, and we even tried to get at it. Carl Mose has passed away, and the culture collection had been continued, but a colleague of mine who's familiar with the site has told me that that is no longer the case, and this was a few years ago. So this was done in 1981, well before a lot of advances in sequencing, so we don't have the diversity. And so that, again, I just want to make a point there, not to be, we presume that these are more diverse in a phylogenetic sense, but we don't have direct evidence. It's certainly larger in scale. So the scale is larger, but we don't have direct evidence from the phylogeny. Yep. Actually, we think probably a lot of them are fast-growing vibrios or related heterotrophs given the culture conditions. Yep. Yep.
When you talk about coexistence, that's like a very ecological question. So I was wondering, how do you link that you see that species are coexisting into an evolutionary consequence? Like, if you achieve coexistence, I mean, they're all stable in the system, they're all there. How does that become a selective pressure eventually?
I see. Okay. So maybe I should explain, step back a little bit. Here I've assumed already this network is in place and said, if the network is in place, what would the trade-offs be that would allow it to be present? And I think you're asking, what is the generative process by which coevolution could lead to that space? In fact, that remains somewhat of an unanswered question. We have worked on coevolutionary models where you start with a low diversity environment. And you can imagine, similar to this, that a resistant host arises, but with some cost that means that now you have 2 bacteria and 1 virus. If I play this game further, then you could have a diversification event on the viruses where now a virus could specialize maybe on this new type or have a host range expansion. So in simulations, we certainly can see that you can start from low diversity and evolution can bootstrap itself up to a high diverse or a highly diverse assemblage. We've done similar experiments with Justin Meyer, we're analyzing some of them, where you actually see this diversification happen. Though what's interesting, at least in the experiments, the coevolution experiments, they often end at some point, whether it's because of size or again because of the way that Paul Rainey described, we're not doing horizontal transferring. So within this particular flask that's being transferred, a super resistant type emerges, and at least in the particular environment, there aren't enough costs to it, and it then gets rid of all the viruses. So I think there is still a gap between these microevolution experiments and clearly the diversity that's sustained at environmental scales. Yeah. Yep.
So when people build these networks of viruses and hosts, it's usually by one trials. And I was wondering how community context would interfere with those results, and how that idea of doing the reverse engineer that you're proposing could help illuminate some of these community context effects.
So when you have to clarify, community context meaning you think there's 3 body interactions, or because in a different, or because the resource, maybe clarify, I don't know.
Yeah, like multiple interaction partners, like multiple phages infecting the same bacteria might have a different result than when you do one-by-one experiments.
So another excellent question, in the sense that all of these phage bacteria infection networks are pairwise. They're 2 body interactions. And we do not know what is going to happen in a natural system when you take, there could be priority effects where you have 1 bacteria, 2 viruses infect, and one is the better intracellular competitor. We're asking right now, does that 1 virus infect that particular host? And we can answer that question even if there are community effects. So I think another way to ask your question is say, will community effects, how important are they? Or can we, if we were to do co-infections, first of all, do they happen frequently enough for these lytic types that makes a difference? And my instinct here is that the priority effects that I'm most worried about are between chronic or latent viruses and lytic viruses, because I think those are going to be the kinds of 3-body interactions that are going to happen frequently, and we just don't know yet. We see examples of co-infection, but no one has done a systematic 3-body study to also see, in some sense, virus with respect to 1 host. If we pool a bunch of viruses that all can infect the same host and we co-infect them, which one has priority? That would be cool, but I don't know of any study that's done that. Yeah.
Can I? I'm sorry. So you have all those nested matrices or interaction matrices.
Yep.
Would you believe that this is like the steady state or the equilibrium state of the interactions? Would you expect like different shapes or topologies of the matrices through the evolution?
Yeah, so I think we do expect it. In fact, in the model that I didn't talk about now, we tried to say, what would be conditions that you could get into a kind of a moving steady state of these nested networks? Yeah. And we found that depending upon certain costs, we could actually drive the system into a modular state and have that to be the sort of steady state. So at the moment, I mean, part of what I'm trying to do here is to show you the empirical evidence, but not necessarily to tell you that we have all the explanations for why they arise. So certainly at small scales, we see nestedness emerge again and again. In evolution experiments, it seems to happen repeatedly. Multiple groups, different kinds of systems. And that raises some questions. Is that something about fundamental biophysical trade-offs or biophysical constraints, or actually trade-offs? And my tendency is actually just trade-offs, that there's some trade-off involved, and that's why we keep seeing it in different systems, rather than there's some biophysical limit to which bacteria can infect which virus. I think there are often trade-offs, and the trade-offs then give selective pressure to generalize or be more defensive, leading to this nested effect. So I think there will be conditions like a strong antagonistic coevolution that are going to favor nestedness, but in natural systems, it has to be modular at some scale. It has to be, there's just too much specialization.
Yeah, and I have another question in this same, I don't know, direction, but I don't know if it's the same. So if we start with, I don't know if it's possible in the lab, if we start to evolve with 2, 1 host and 1 pathogen, 1 virus, and how would you expect that the shape of the matrix, because then the many types of viruses and hosts will appear, would you expect that the shape would evolve like?
Okay, so I can answer that at least to some extent. When you saw those nested interaction networks, some of them from evolutionary experiments in which the generalist viruses appeared later in the coevolutionary process. That's and the most defended kind appeared later too. So there was actually a directionality there, and we've redone some of these experiments, again, with Justin Meyer, and that is the tendency. So there tends to be a directionality you could think of in the rows or columns, towards host range expansion on viruses and becoming more defensive amongst the bacteria, because keep in mind that there's a selective benefit to be resistant to all the viruses that are present, which tends to move. It turns out that often in cases, resistance to the current types tends to confer, and that is partly unsolved, resistance to the prior types as well.
So the diversity of viruses increase first, and then the diversity of hosts increase.
I don't think you should think of them necessarily as being one happens then the other happens. They're both happening at the same time.
Okay, cool.
Yeah. And in fact, I think we're probably undersampling. I know we're undersampling the extent to which the diversity is happening at the same time rapidly. Yeah, we've tended to look at these, which ones we can measure. Like I showed you, total bacteria or the one resistant kind, but when we've done deep sequencing, there's many more kinds that are lurking very early in the experiments.
So yeah, I was just wondering, because of those cycles that you said they were like in different direction, if you would like say how diversity would emerge first.
Yeah, and that experiment, they didn't do all the sequencing. I don't know all the different types, but that's a case where there were oscillations rather than directionality. But in the other experimental evolution work that I've analyzed, there is a sense of directionality.
Okay, cool.
Yeah.
Thank you.
A sense, yeah.
What about the sizes of the complex communities? It seems like you have a full range, from the big module as very small module, and I thought there was like an intermediate range where things could work out. Can you comment on that?
Yeah, so I think we should keep in mind that what we do in the laboratory is a means to try to understand far more complicated natural systems. And so with respect to tractability, a lot of these experimental systems are 20 by 20. When you start to talk to your experimental colleague and tell them to do 50 by 50, they're not happy with you, right? For good reasons. And it's not even clear that you're going to learn that much more. On the other hand, the natural systems are extraordinarily more diverse. And so at present, the idea of going from these methods that go from 10 by 10 to 1,000 by 1,000 or more is not feasible. So as I've said, that's part of the reason we think it's important to at least begin to poke. And I think that the thing that we're trying to do is to get at least a sense of the community interaction networks amongst relatively dominant players. And players could be by abundances or by function, contributions to, let's say, regeneration of nutrients or lysis, which may not always be the most dominant microbes, because maybe a bigger player for mortality is a less dominant microbe because it's being killed by viruses, right? Or a rapidly turnover kind, which numerically is not so dominant, but is a major player. So I'll just turn your question into something that I think I'd like to see the field go, which is to do infection networks from an unbiased perspective, culture-independent perspective, but prioritizing those partnerships that are relatively important ecologically. So if we can get there, and I don't think the answer is going to be it's thousands or tens of thousands when we start to focus in on certain functions.
So I'm still worried about this nettedness. I mean, you found this nice ordering when things are not that complicated. But then you use it also when you have a lot of nodes and a lot of columns. And might it not be that like you're using this when you maybe like stuck in a local minimum, which is good for one thing, but then in some other thing, something else may be better? Yeah,
so first of all, I want to point out, it's an excellent point because actually the large-scale network annoyed us for many reasons, not least of which is that some of these are heuristic algorithms in some cases, but there are other ways to calculate nestedness that are independent of the heuristic. So I'm using the heuristic to visualize, but using a different method to do the calculation. Nonetheless, if you have a network, the nestedness algorithm essentially assumes that you can place things in the upper left component. You can imagine a modular network which the nestedness algorithm thinks is nested because when you put two modules, there's disproportionate number of components. But really, the structure is dominated by modularity, and then there's nestedness. And so we've been trying, we haven't successfully done it, tried to figure out a way to hierarchically structure one of these bipartite networks by comparing at the community scale, is it more modular, is it more nested? Once we make that decision, then zoom in and keep asking those questions. So in the case of the large network, I think we're confident that it's a modular network. With respect to the heuristics, you're right, there are a couple of ways of calculating it, something called NODF and temperature calculator, and we tend to use those, the temperature calculator for visualization, but a different way that is more robust for actually calculating the nestedness. And that's just comparing exhaustively all pairs and asking about nestedness, so it doesn't depend on the heuristic. Yep. We're almost done with, I think.
So we're going to have more questions after because people have to be back for
Correct.
Okay.
So let's say at 3:45 after the colloquium, we'll reopen for questions.
Or I can just also hang out after the colloquium and just chat it over coffee, whichever is easier.
So we have an hour after
Okay,
the colloquium until the next seminar.
Okay. All right, thank you.
Thank you. Figure 8 cycles? I'm sure you can, but
We haven't